So let's talk about the Sanger method, or chain termination method of sequencing. It was developed in 1975 by Frederick Sanger, but it's still widely used today, particularly for cloning applications. So it relies on basically DNA replication. So you need a polymerase, and you're going to use it to amplify some template sequence. And so you need all the stuff that you need for replication. You need your deoxyribonucleotides, you need your template, you need a primer, and you need your polymerase. But there's a slight modification in that besides putting in just all the regular deoxyribonucleotides, you're also going to put in a very small fraction of dideoxynucleotides. And so what does that mean? Let me show you a picture. So over here on the left are regular deoxyribonucleotides, and they're called that because there's no hydroxyl group at the 2' carbon, right? So that's why they're deoxyribo. And that's in contrast to ribonucleotides, which would be used for RNA synthesis. Now, dideoxyribo also don't have this hydroxyl group here, but they, in addition, don't have a hydroxyl group attached to the 3' carbon. And so you really can't use these as a substrate for the addition of the next nucleotide. And so this guy can be incorporated into a growing DNA strand, but once it's there, you can't add another nucleotide here. So you cannot add the next nucleotide. So they're basically chain terminating nucleotides because they don't have the proper group on that 3' carbon. And so what happens as a result of this? So first, let's take a look at normal polymerase reactions. So we've got our template strand. Then we're going to denature the template strand so that it's single-stranded. And we need to add a primer. And we actually are going to use a labeled primer because we're going to want to actually visualize our DNA after we're done synthesizing it. And so we use a radioactive label in order to be able to do that. And so we put in a primer that already has a radioactive label on it. We put in our polymerase and our dNTPs, and we end up with, new DNA, a new double-stranded piece of DNA that has this radioactive label on it. So that's great. That's just normal polymerase reaction. Now, what about if we have all the same components in here, but then we also put in just a small fraction of these dideoxyribonucleotides? And so what happens then? So we still have our polymerase. We've got all of the regular nucleotides. And now we're going to put in just a small fraction of either dideoxy-TTP, dideoxy-CTP, dideoxy-ATP, or dideoxy-GTP. And so first, let's look at the dideoxy-TTP. So what will happen is it will start amplifying, and occasionally when we're going through this amplification, like let's say, we're going to start over here with this A. Maybe one of the regular DTTPs is going to come in and base pair with that A, in which case the amplification will continue. Then we make our way to the next A, and maybe, again, a normal guy is going to come in, and so our amplification will continue. And then we make our way over to the next A, and then because there's a small fraction of these dideoxy guys, maybe that guy is going to actually come in to that last A, and it's going to actually cause a termination of the replication at that spot. And so in bulk, since we have multiple, many thousands of millions of strands of DNA in there, we can expect that we're going to get a mix of product where some of it is going to terminate right here, some of it is going to terminate here on the other side of that A, and some of it will terminate here. And that's actually what we see. So this top guy made it all the way to the end almost, but it terminated at that last A. This guy terminated at the second A, and this one terminated at the first one. And so we should see a mix of all three of these different lengths. And those lengths then correspond to the last place where we incorporated a T. And so for the dideoxy-CTP, it's the same thing, only now we're looking at incorporating these C's where we have G's. And again, you're going to get all the different lengths that you would expect that are complementary to that dideoxy-CTP. And the same thing for the dideoxy-ATP, same thing for the dideoxy-GTP. And so if we run these on a gel right next to each other, so we load them all on a gel, then we would expect to see something that looks a little bit like this. So here are all our dideoxy-TTP reactions, CTP, ATP, and GTP. And so we can kind of see that these should all be different sizes, right? So you shouldn't have anything in, the first lane that's the same size as the second lane, just because of the way that this reaction works. And we can actually then predict what the sequence of that DNA was by looking at the lanes and saying, okay, well, the first guy in our sequence must have been a T, because that's in the T lane, right? The second guy was what was that? An A. The third guy was a T. The fourth guy was a C. And so we can use this to sort of reconstruct what the original sequence that we amplified was. So we have T, next one was A, next one was T, next one was C, and then another C, and then an A, and so on. And that's essentially how the process works. And so if you actually look at one of these gels, it looks something like this. And so here's the gel where this is actually in practice, or shown in practice. And so if we take a close look at this, we can actually determine what the sequence is that was used in these reactions. So the first nucleotide was a G, then they had another G, then we have a T, then we have a C, then an A, then I think maybe a T, then another A, G, C, T, G, and then TTT. And so we could just read this gel all the way up and figure out what our original sequence was based on where these bands are. And one thing to think about then is, as you get up towards the top of the gel, resolution is it's a lot more difficult to resolve between the individual nucleotides. And that's just because of the way electrophoresis works. So the further down the gel, you get better separation. And that's just always the case. And so how many nucleotides do you think you'd be able to accurately sort of identify by looking at this gel? You don't, you could guesstimate what you think. So now I'm going to talk about a massive improvement to this method. And it's really kind of a simple sort of change, but it really made it easier to do this type of sequencing. And that was to replace the radioactive label on the primer with actually fluorescently labeled nucleotides. And so instead of having our end of our guy radioactively labeled, we would just make each of the nucleotides a different fluorescent color. And then when we run the gel, we could see that, our A's are purple, or our C's are blue, and our T's are green. And what is this? That's blue also. I guess maybe that's supposed to be the G. I don't know. Anyway, the point is that they're all different colors. And you might say, well, how does that change anything? Because now they're just, okay, they're easier to look at, but you still basically use the same process in order to be able to read the sequence. But what this allows us to do, having them different colors like this, allows us to, instead of running them in separate lanes, allows us to run them in the same lane. And so now we don't have to read across 4 different lanes to try to figure out the sequence. We could do it all just in one lane. And then we could tell the sequence based on the color that's coming up next. And in modern facilities, this is actually done not just on like a regular gel. It's done in these little capillaries that have polyacrylamide within the capillary that allows us to sort of resolve these guys to a much greater extent, and also uses less material, and it's just a little bit easier to actually work with. And so this is kind of the readout that you would get from running one of these capillaries. So each of these peaks represents the color that is indicative of whichever nucleotide it is that was attached next. And I know if you work in a lab, you've probably definitely seen something like this before. And what you're really looking at is essentially the equivalent of what we have on the left here. So we have, except for in like a large gel, and instead of the DNA being radioactively labeled, we have this in a small capillary tube, but the same sort of electrophoresis process in a small capillary tube, and each of those nucleotides being labeled with a different color fluorescent dye that allows us to get this type of readout. And so the Sanger sequencing method can give us accurate reads from between 700 to 1,000 nucleotides. You don't need a lot of the DNA to start, but you do need to have some of the, you wouldn't use this for a whole genome or anything like that, because you do need to have like relatively pure sample of DNA. You can't throw in just a bunch of different samples. If you did, you would get like a huge mishmash of data, which is all overlap.